In this presentation, I'm going to talk about how RNA interference or RNAi works. So first, what exactly is RNA interference? Let's talk about the different types of RNA. The first one is mRNA or messenger RNA. Its job is that it is an RNA transcript of DNA to be translated into protein. It is complementary to the DNA, and this is where tRNA comes in. tRNA is transfer RNA, and it is involved in translation. It carries an amino acid to the ribosome, which brings us to the next one, rRNA or ribosomal RNA. It forms the ribosome itself, and it has enzymatic function. So RNA is special because it itself can act as an enzyme. Fourth, we have long non-coding RNA, and what it does is it regulates gene expression. It's pretty similar to RNA interference, but it's not exactly the same. One example is imprinting and imprinted genes. Next, we have small nuclear RNA or snRNA. It aids in splicing. And lastly, we have siRNA and miRNA, which are small interferent RNAs and microRNAs. And those are involved in the regulation of gene expression, or more specifically, gene knockdown. Now let's talk about the central dogma, which is the process of DNA, mRNA to protein. So DNA is transcribed into mRNA, which is then translated into protein. There's a double arrow between DNA and mRNA because of reverse transcription. And therefore it makes sense that if there's more mRNA, which is the substrate, then there would be more protein, which is the product. Because of Brownian motion, which basically says that molecules move randomly in the cell, there will be a higher probability of a ribosome attaching to an mRNA and translating it into a protein. So in the 90s, plant biologists were trying to make petunias more purple, so they decided to add more mRNA in. So you would think that with more mRNA, there would be more protein, and therefore the purple color in petunias would be darker. And that's exactly what the plant biologists thought. But instead of making it more dark, it made the petunias white. So why was this the case? That was because of RNA interference. So this brings us to the discovery of RNAi, which was in 2006, where Fire and Mello got the Nobel Prize in Medicine. So first, this is an experiment that they did. So the RNA in this example codes for a muscle gene. So if there's no mRNA present, the worm will twitch. And if there's mRNA present, which means normal function, it will not twitch. So in the sense RNA, which is basically just this transcript, injecting it was wild type. Injecting more mRNA left the worm normal. So there is that mRNA present. Adding the antisense RNA yielded the same result, but adding double-stranded RNA changed it because it took out the mRNA. There was no mRNA, so the worm twitched. So what is so special about double-stranded RNA? This relates to the petunias example because in that case, biologists also injected double-stranded RNA instead of single-stranded RNA, which knocked down that gene. So this is the mechanism for siRNA. So first, siRNA enters the cell. It's important to note that it's foreign. It's not made by the cell itself. So if it's too long, Dicer processes it and makes it around 20 to 21 nucleotides in length. Then RISC attaches to it and gets rid of one strand, the passenger strand, which is shown here. And whenever this RISC factor combined with the RNA collides into mRNA, a target mRNA, which is complementary, it will deliver a double-stranded break if it's perfectly complementary because it has argonaute in it, which is an endonuclease. If it's not perfectly complementary, then it will just prevent translation. Now let's talk about miRNA or microRNA. So the biggest difference is that it's endogenous, unlike siRNA, because it's made by the cell, it's not a foreign thing. The mechanism is mostly identical, and this is a way for eukaryotes to regulate gene expression at the translational level. So first, since it's endogenous, it's from the genome itself. So a part of the genome is transcribed into miRNA, which is a hairpin structure. And then it's processed by drosha, which just clips the ends and makes it a hairpin, which then it exits the cell. And it basically now becomes a siRNA because first Dicer processes it, which turns it into a double-stranded RNA around 20 to 21 nucleotides in length. And then RISC gets rid of one of them, the passenger strand. And RISC and one strand of the RNA go around the cell, and when it collides into a target mRNA that's perfectly complementary, Argonaut will deliver a double-stranded break because it's an endonuclease. And if it's not perfectly complementary, if it's a little bit complementary, slightly, then it's just going to inhibit translation. It stops translation, so therefore it regulates gene expression at the translational level. It's also important to note that taking out the mRNA also prevents translation, so technically it's still post-transcriptional or translational level gene expression regulation. Now that we've established how RNAi interference works, let's talk about its use in medicine. First, let's talk about gene knockdown versus gene knockout. Gene knockdown is controlling the RNAs, the mRNAs usually, in the cell to prevent the production of proteins, whereas gene knockout is actually taking out a portion of the genome. An example of gene knockout is CRISPR or restriction digest, which is very different from gene knockdown. And in medicine, we prefer to use knockdown instead of knockout. Well, CRISPR is becoming more popular, but it's not perfect yet because there are a lot of off-target effects and drug delivery is another issue. So the goal for this is to reduce production of abnormal proteins, and there are only 3 RNAi drugs that have been approved by the FDA because it's so difficult to develop. Some problems are off-target effects and drug delivery, which are many problems that are very similar to gene knockout processes like CRISPR, but because it's just a molecule, it's a macromolecule, and you need to make sure it just targets what you want, and you have to get it inside the cell. Now we're going to talk about all 3 drugs. The first drug we're going to talk about is patisiran, which is a treatment for ATTR amyloidosis. ATTR amyloidosis is a fatal rare autosomal dominant disease, and symptoms don't appear until people are adults. And patisiran treats disease by targeting TTR mRNA. Since it's a siRNA, it's a RNA interference drug, its goal is to prevent fibril buildup by reducing the mRNA and thus the protein. So it does gene knockdown because it takes out the mRNA and not the gene itself. So go into more detail about ATTR amyloidosis. So the definition of amyloidosis is the buildup of mutated proteins in the body that resist catabolic processes. So what does that mean? So proteins that are misfolded, they have the wrong amino acids, are in the body and they're bad. Some of them are fine because they can be broken down, but these mutated proteins can't be broken down by catabolic processes, so it's a problem. And the problem with this is that for this case is that it forms these fibrils, which are pretty bad. So normal TTR, so patients without ATTR amyloidosis, their TTR forms a tetramer. But those with ATTR amyloidosis, their mutant TTR destabilizes the tetramer. So it turns into amyloid fibrils instead of the tetramer. And in different people, it can present differently, but in most cases, it's cardiac or neuropathic, which means it most likely affects the heart or nervous system. Second is gavosiran, which is a treatment for acute hepatic porphyria. So it targets the LS1 mRNA hepatocytes. Since it is also an RNA interference drug, it reduces the expression of that protein by targeting the mRNA, which is, again, gene knockdown. So LS1 is a protein that plays a role in the heme production in the liver. Going into more detail about acute hepatic porphyria or APA, it's a really rare genetic disease that's pretty life-threatening. And APA occurs when there's a problem in heme production, which is hemoglobin, which results in the production of PBG and ALA, which are toxins. So, in normal heme production, there's low levels of PBG and ALA, but in people with APA, it produces a lot of these toxins, which is pretty bad. And symptoms are often unpredictable, but the most common ones include seizures, fatigue, muscle weakness, and tachycardia, which is a really fast heartbeat. So therefore, targeting the LS1 mRNA reduces the production of these toxins levels circulating in the bloodstream, but also reduces heme production. The last drug we're going to talk about is lumasiran, which is a treatment for primary hyperoxaluria type 1. It targets this enzyme called glycolate oxidase, which function is to turn glycolate into glyoxylate, which is the production of oxalates. So targeting this enzyme or this mRNA results in less oxalate production in the liver, or hepatic oxalate production. This disease is really rare and it can cause end stage renal disease, which is fatal. And similarly in kidney stones, it's also caused by a buildup of oxalates in your kidney. And in this case, the liver does not create enough protein to prevent the overproduction of oxalates, which causes oxalate buildup in blood vessels and organs, which can cause kidney stones. But this is a lot more severe because this can cause end-stage renal disease, which is really bad. And symptoms include pain while urinating, severe back pain, and blood in the urine, which you notice that it's very similar to kidney stones, the symptoms itself. And this brings us to the end of this video. Thank you for watching.